Hello, biology class. This is Miss Jeannie and we're talking about Lab 11, the results of Lab 11. I'm going to start on page 53 where you have your questions. That's the lab that we did today, the bacteria lab. Number 1, compare the size and shapes of the 3 different bacteria types, Coccus, Bacillus, and Spirillum. Which is the smallest, which is the largest, and how do their shapes differ? Well, our answer key says that the Coccus are the smallest and the Spirillum are the largest, although they were pretty darn small on our samples, weren't they? They were all pretty small. And the 3 forms of bacteria are named for their shape, so they all have different shapes. The Coccus being round or cylindrical, the Bacillus being rod-shaped or straight, and then the Spirillum being little spirals. How do the bacteria, number 2, how do the bacteria groupings correspond to their name prefixes? Well, some bacteria grow singly, and others form colonies of cells that display different groupings. For example, 2 cells that group together would be called a Diplococcus or Diplobacillus. Di meaning 2. If the cells form into clusters, they take the prefix staphylo, like Staphylococcus. And if they form filaments or strings, they're kind of stringy, they have the prefix strepto. Like if you had strep throat, that's the prefix strepto, Streptococcus, Streptobacillus, that type of thing. Number 3, how do Staphylococcus and Streptococcus differ? Well, staph is grouped in clusters, and Streptococcus are rod-shaped and grouped in strings. So you can have a coccus, which is a round bacteria, end up looking rod-shaped if it's a Streptococcus. Number 4, why is bacteria viewed and drawn under high power instead of medium power? Well, we know that because they're so darn small. Right? They were not seen very well under low power. We had trouble finding them. So that was page 53. I'll show you a picture of page 54, which I did show you in class today, but just in case you don't like your drawings, you can take a look at these. Make sure your drawings are labeled. Here's an example of spirillums. They look stringy or spirally, like little snakes. And that's at high power.
No, not really.
Here's a picture of Bacillus, the rod-shaped, also shown at 400. And here is a picture of the Coccus, which are the little teeny round ones, also at 400. But I think we looked at those. I think we had a good view of those today. So for homework, please do not forget to start the bacteria part 2 lab by preparing your petri dish tonight, or your culture dish, so that you're ready to swab your teeth tomorrow morning to get the bacteria off before you brush, after you brush, and after you use the mouthwash. And then starting Tuesday, you're going to want to go to, let's see what page it's on, page 59, table 12.1, to start putting in your results. You know, do you have any growth? What does it look like for all of the different quadrants of your culture dish? And then that goes on for 8 days. Please check it every day and record it in there. And then the 8th day, I think, will be next Tuesday. So you can just give me the results on Monday. And if I get any good pictures or anything of my girl stuff, I will put it out on the web for you to see. Additional homework is reading the next 2 labs. Lab, I think it's 12. Let me look. It might be 13. No. Lab 13, which is the classification lab. I'm going to assign that to you for homework next week. You're going to classify some types of beans, and we'll do the Protista lab, which is lab 14, in class next week. So you guys have a great week, and I'll see you later.